So I'm very pleased to introduce the closing keynote speaker for Restoring the West 2014. It's interesting as we work in comparable career paths with a lot of people, we meet people and we think maybe this is just a flash in the pan, I'll meet this person, never see him again, maybe never talk to him outside of this conference. And about 7 years ago, I actually was given Heida's name by my then master's advisor, and he said, this woman, she does everything. You need to call her. Whatever you want to do, she's done it, and she can do it better than you. And I was like, wow, I need to call this lady. Let's start the slideshow while I'm dragging off. So I called Heida, and she spent like 3 hours on the phone with me talking about everything from eelgrass to the lower Columbia estuary to tidal rivers to large woody debris to geomorphology to what it was like working with Dave Montgomery. And through the conversation, I realized not only is Heida a world-class scientist and an expert in a field full of awesome scientists, but she's a very, very generous person. She's a very, very mentoring person. And when she says something, she really means it. And I'm really excited that she's made it out because she works at the Pacific Northwest National Lab, which is a very prominent organization, but not very well known for the great ecology that they do. So Heida has worked on many, many projects, and she's going to explain to us how we can kind of see the forest, individual, or many, many projects across this watershed, as well as these individual projects, kind of the trees. And she'll do this using kind of an ecological and geomorphic approach. And I'm very, very happy to introduce Heida Diefenderfer as our closing keynote for Restoring the West. Let's welcome her.
I don't know how on earth I'm going to live up to that introduction. I feel like I won the lottery having the hour-long slot, and I'm hoping to make good use of every minute of it today. Thank you all so much for staying for the last hour of this conference. Like Mike, I feel like it's just been a wonderful couple of days. I've learned so much, and I truly am honored to have an hour-long slot when I'm in the company of such esteemed scientists. So this is really fun. First, I'd like to thank my kind of little band of brothers, my team that I've been working with for the last 10 years, Amy Bordy, Gary Johnson, Ron Thom, and the four of us are out at the Marine Sciences Lab, and then Andre Coleman from the Hydrology group over in Richland. And I want to especially highlight Andre's participation because he is a USU grad from 1995 from the College of Natural Resources. And he went on to do a master's degree at the Free University in Amsterdam in geoinformatics and has just provided incredible contributions for us in terms of landscape scale data processing and integrating hydrology and geology. And so I know that you guys have wonderful programs here because of working with Andre for the last decade. I also need to highlight some other key collaborators. I assume that Nate is referring to Kern Ewing, and so these are the members of my doctoral committee, Kern and Dave, John Skowski and Christina Vogt, and some of what I'll be talking about today is from my dissertation. And then David J. and Keith Leffler from Portland State, and Curtis, Lori, and Lyndall from the National Marine Fisheries Service, who've provided really invaluable fisheries perspective on our work. Ian and Scott from the Columbia Land Trust. A lot of our work has, sorry, can you not hear?
We're going to turn you up just a little bit.
Okay.
You're good.
Okay, Ian and Scott from the Land Trust, and some of our work has been on their sites, and also they've translated some of our findings into new restoration project designs. And then finally, Keith and Gina from the Lower Columbia Estuary Partnership. So today I'll start by talking about the study area and the target fish species and the Columbia Estuary Ecosystem Restoration Program and our research objectives and approach, and then give some key results at the estuary scale in hydrology and vegetation, and then do a little bit of a dive into the Sitka spruce forested wetland case study. I added these slides yesterday after kind of perusing the posters and realizing the deep interest in geomorphology here, and this is kind of a geomorphology chunk on the Sitka spruce. And finally talk about the evidence evaluation of the cumulative effects of large-scale ecosystem restoration. So these are some of our study sites here. At the top left, you can see kind of approaching a Sitka spruce swamp, a typical spruce swamp at dawn. On an island in the lower Columbia. And at the top right, this is a brackish marsh, also in the lower Columbia. And so I'll start by talking about the study area, restoration program, and research objectives in this section. So this is the Columbia Basin, which you're all familiar with from other talks in the last couple of days and your other prior knowledge. But we do have 13 ESA listed threatened and endangered stocks of salmon and steelhead in the Columbia right now. And their natal streams really range all the way from the Snake River to upper and middle and lower Columbia to the Willamette. And we have 32 major dams, which the operation of which really affects our study area at the lower river. This is a figure from David Jay, the geneticist for the National Marine Fisheries Service, who is the guy who analyzes your fin clips. And in the estuary, we have an interesting problem where if you take a grab sample of salmon, you don't know where in the basin they're from. We have salmon outmigrating and steelhead outmigrating year-round every day of the year. And so sorting out the stock, the origin of stock, is a very difficult problem here. And it ties into this kind of complicated early life history diversity that we've begun to unravel, or scientists, especially at NIMS, have begun to unravel in the lower estuary. We're learning more and more that there are these significant tidal freshwater and estuarine rearing stages for some of these stocks, or under certain environmental conditions, which might pertain in a certain year but not the next year, et cetera. So this talk is really focused on the outmigration period and the estuary rearing period. And I did want to note that NIMS, in their biological opinion, includes tidal freshwater as part of their estuarine portion of the basin, which is not something that most estuarine ecologists would do, but we've lived with that now for the last 15 years. So the program driver for the Columbia Estuary Ecosystem Restoration Program is creating habitat, restoring habitat for juvenile salmon. And really it began in 2000 because there had been multiple NIMS biological opinions before that that didn't include the estuary as something that was important for survival. And the first one that did identify that as important was in 2000. So this is the lowest major dam, the Bonneville Dam, which is the head of our study area. It spans a couple of river kilometers. And you can see it here just north of Mount Hood and southeast of Mount St. Helens and upstream of Portland. And some key characteristics here, this is a recent historical floodplain delineation by Jim O'Connor at USGS in Portland. And it's about 1,500 square kilometers. Salinity only ranges, the estuary proper really only ranges up to about here. And the head of tide is at Bonneville, but the tidal range down here is 3.6 meters, whereas it's very small, just a matter of inches up at the dam. In the red here is the diked area, and this is really the crux of the matter. The other colors are the tidal wetlands, forested wetlands, scrub wetlands, and herbaceous wetlands. And we've got these barriers to flow, fish passage, and tidal wetland habitat function throughout the historical floodplain, mainly tide gates and culverts through roadbeds and dikes. These are our 5 wetland plant communities, although I'm starting to really think about guilds in the last couple of days. May need to do some more work on that. But we have the brackish marsh here, which is in the lower part of the estuary, longitudinally speaking. And then tidal freshwater marsh, which is above that 20 to 40 river kilometer range. And then scrub wetlands we have throughout the estuary at the elevations that are just a notch above the high marsh. And then we have 2 types of forested wetland, maybe 3 depending on how you call it. We have in the lower part of the river, kind of congruent with the coastal fog belt, the lower 60 kilometers or so, we have Sitka spruce swamp. And then in the upper part of the river, we have riparian forested wetlands dominated by ash or cottonwood. And I know that a lot of folks here are probably really interested in the willow and the riparian forested wetland, and I'm afraid this is where we have the fewest of our sites. We've sampled 60 sites in the last 10 years, and we've really focused on reducing the uncertainties about the tidal freshwater and the Sitka spruce swamps. So the kind of habitat restoration that's being done is hydrologic reconnection of the main stem river with the habitats lateral to the main stem river and islands. And so this includes tide gates, culverts, and dike breaches primarily. There's not a lot of dike removal because of the risk factors, which is unfortunate because this really is a gradient of hydrologic connectivity here. So I'd like you just to visualize for a moment, go back in time to 2003 with me when we were brought in by the action agencies, Bonneville Power and the U.S. Army Corps of Engineers, to try to help them respond to the fact that in 2000 the NIMS had added the estuary to their overall program for Columbia River restoration. And we had to ask ourselves, how do we implement and evaluate a large-scale ecological restoration program to recover economically and culturally critical fish species in a complex ecosystem that we know relatively little about, which encompasses these extreme aquatic, terrestrial, and tidal fluvial gradients. The thing that's interesting about this is that this study area was effectively created by the biological opinion in 2000. Nobody had ever researched this particular geographic area as a whole that we know of, because the estuarine scientists have been working down in the estuary proper where there was salt, and then we had some studies by the state agencies, the natural heritage programs in Washington and Oregon on their respective sides of the upper river, but nobody had really considered this thing an entity until the biological opinion called it the estuary. So we had a little bit of guidance because the opinion itself said that we should develop and implement a methodology to estimate the cumulative effects of habitat conservation and restoration projects in terms of cause and effect relationships between ecosystem controlling factors, structures, and processes affecting salmon habitats and performance. So that wouldn't be too hard, right? So in a nutshell then, our research challenges for the last decade were to develop an evidence-based evaluation system for large-scale ecosystem restoration, to concurrently reduce the uncertainties in the fundamental knowledge of the ecosystem, and to help the agencies prioritize, implement, and adaptively manage the restoration program, and hopefully not to be too far wrong, because we do agree with the biological opinion that this region is very important to these fish. So let's talk now about the process we went through to develop the research approach and the adaptation of evidence-based evaluation methods from other fields for restoration ecology. And these are some of the elements of the work that I'm going to be talking about today in black, and some of the parts that I'm not going to talk about today in gray. So a few highlights coming up will be the ecosystem conceptual model, monitoring indicators and protocols for this newly defined region. And then in the terminology of the BIOP, we did status and trends research, critical uncertainties research, and action effectiveness research. And I know you're all familiar, or some of you at least are familiar with those terms from the BPA Fish and Wildlife Program as well. And then again, highlighting Andre's work, I'm going to focus a little bit on hydrogeoinformatics and modeling of inundation and synergy. So as just a word to the students in the audience mainly, this type of work really requires a transdisciplinary team. And by that I mean that we can't keep the disciplines separate. We really have to forge something new, or at least do our best to address these kinds of problems, especially in our case where we were really drawing from both estuarine and fluvial sciences. And this is kind of an idealized restoration trajectory based on the Bradshaw 1987 model that shows a projected correlation between ecosystem structure and function, with a fully developed ecosystem here, a reference site type ecosystem, which I'm going to be using that term a lot, up at the top right, and then an early development or high disturbance system down at the bottom. And in our system, we also have some sites that would hypothetically be somewhere in the middle. And these are sites where there were accidental breaches decades ago, and also sites where there's been habitat created through dredged material deposition. So I'll be talking about some of that as well. So the evidence-based evaluation, I'm sure some of you have run across this type of work in your work, but I'd like to talk for just a second about the foundations here in prior research. In the literature, it's been characterized as a method of evaluating a collection of published, peer-reviewed literature to determine the weight of evidence for or against a hypothesis. It uses causal criteria, so these are aspects of an association that really should be considered before you claim that it's a cause and effect association. And according to Hill, Dr. Hill in 1965, who proposed it in the Proceedings of the Royal Academy of Sciences, none of those causal criteria can really individually prove the case, but all of them are indispensable. This really originated simultaneously where it was also included in the report of the Surgeon General, which determined that smoking was harmful to your health. And you can imagine that despite the very large weight of evidence that had been funded by the tobacco industry showing that it was not harmful to your health, they actually came to that conclusion using this method of causal criteria. It's really a critical thinking-based method, and it's trying to separate the wheat from the chaff in all the data that's out there. It was further developed then in epidemiology and ecotoxicology, and has recently been concurrently applied with our study by some folks over in Australia. Dr. Norris has passed away, but also Angus Webb and Joe Greet. Okay, so I'm going to spend a little bit of time on this because this is really kind of the product of our overall approach that we developed, and most of the rest of the talk will be addressing pieces of these results that respond to this framework. So it starts at the bottom with a kind of typical ecologist approach. Let's make an ecosystem conceptual model that really sets down the state of our understanding of the ecosystem, and then draw from that a hypothesis framework and monitored indicators. But where it starts to depart from that is with the lines of evidence here. And we have developed 7 lines of evidence that we think may be applicable to other ecosystems as well. We hope they are. They should be fairly generalizable. One is this evidence-based review of the literature. So this is what evidence-based evaluation is in the literature to date. Then we have 5 that are based on the floodplain. The evidence-based review is global, right? So it includes all the analogous ecosystems where similar restoration has been done globally. So this is a great way to pull in data before you've done anything. The only other one that's not on the floodplain is the change analysis on the landscape setting. So this is where we bring in the watershed. We're working on a riparian zone, on a floodplain. Obviously what's going on in the watershed and the basin matters. So we do change analysis on the landscape setting. And then we have these 5 lines of evidence from the floodplain itself. So understanding of the ecosystem, analysis of data on target species, meta-analysis of restoration action effectiveness, modeling cumulative net ecosystem improvement, and hydrodynamic modeling of cumulative effects. And this is the analysis segment, really. And this is where I think a lot of science stops, because this is what we publish, right? This is what we can defend with our statistics, is our final analyses. But what decision makers do need from us is also to make the effort to synthesize and evaluate and tell them something that's useful, not just a finding, but something that's a little bit more toward the recommendation end of things. And so this is where the causal criteria comes in, where we look at this. These are the strength of association, the consistency of association. Is it biologically plausible? Does it occur along a gradient, like a sublethal to lethal stress gradient, et cetera? And finally, we try to filter these through the types of categories of cumulative effects that we know are occurring in ecosystems. We all know, and I'll have a slide on that next. I want to say that we've published this approach in 2011 in ecological restoration, and we just finished implementing it in 2013 as an early stage evaluation of the Columbia Estuary Ecosystem Restoration Program, because all the projects there are just like 1 to 5 years old. And so that's in a report to the Corps and also in review right now. So these cumulative effects categories were identified by the President's Council on Environmental Quality back in 1997. And they're really just ways that effects can accumulate in the very generic sense. So we have frequent and repetitive effects on an environmental system. This can be called time crowding. Delayed effects, time lags, which of course we see all the time in ecology. Dr. Breshta this morning, we heard about some delayed effects and also some indirect effects, trophic cascade effects. High spatial density of effects on an environmental system. I think this is what Joe Wheaton and Nick and Wally are talking about with these high density clusters of beaver dam starters. Effects that occur away from the source, effects that arise from multiple sources and pathways, and then fundamental changes in the system. We've all heard about and wondered about thresholds and triggers. It's a difficult thing to isolate. So we can see the potential for synergy when we look at these large-scale restoration programs. For example, this is a suite of properties acquired by the Columbia Land Trust, and they're separated by other types of properties, and they have multiple different types of restoration actions occurring on them. So how do we evaluate that? Similarly, here we have a suite of tie gates. This is the Julia Butler Hansen National Wildlife Refuge, and because they have to manage for the Columbia white-tailed deer, they don't have the option to really fully reconnect. They need to keep some slightly dry habitat, but they can at least let the fish in and try to garner some benefits that way. So this is the evidence-based evaluation approach in just a more traditional flow chart. I just wanted to highlight that it's hung on a hypothesis framework. So we need to start with a primary hypothesis, in this case, that the Columbia Estuary Ecosystem Restoration Program habitat activities, restoration activities, are having a cumulative beneficial effect on juvenile salmon. And then we split it right away into secondary hypotheses of the habitat-based indicators of ecosystem controlling factors, processes, and structures showing positive effects, and the fish-based indicators in this case, or target species-based indicators showing positive effects from both the restoration actions and the habitats that are undergoing restoration. And then the answer hypotheses are where we get into measurable metrics. And so, for example, our process-based metrics, the water regime, water surface level regime, the sediment accretion, vegetation similarity, and export to the main stem. And then for fish, the presence, residence, survival, prey, diet, fullness, and growth. And this is where we get to the lines of evidence and the part that I showed you earlier with the analysis and synthesis and evaluation. So there's a lot in here that I know is familiar and has been used. What we've tried to do here is to pull it together into a formal and organized framework. And I think some of the highlights that are new are that it accounts for the cumulative effects categories, takes advantage of critical thinking tools. It allows us to try to do evaluation when we might not have a comprehensive multi-million-dollar model of the system to look for effects on. Like, I mean, we've worked on projects like that with the Upper Missouri, for example. The other alternative here is a really large integrated model system, which can be extremely expensive. And also is never conclusive. I mean, statistics are never conclusive. Models are never conclusive. This is not conclusive either, but it's an attempt to create a framework that increases our chance of being right. So it does include as one line of evidence this comprehensive quantitative review of the literature that we see as evidence-based evaluation being used, which has been used for 5 decades. This is just one extraction of the original ecosystem conceptual model that we did in HTML in 2004 so that we could drill down into it and have all the literature that was associated with each box available to us. And I've extracted it in terms of kind of our framework of salmon habitat capacity, opportunity, and realized function. And so capacity is the factors in the habitat that might support or fail to support the salmon. The opportunity is whether or not the salmon can get there. And then the realized function has some of the metrics that we talked about before of salmon performance. And this is just a classic stressors-based model, stressors acting on controlling factors, on ecosystem structures, processes, and then functions. So again, this is the beginning of the process then where we're drawing functional hypotheses from the conceptual model, which we're all doing all the time. I heard this in most of the talks in the last 2 days. So, and these are classic. We're saying there are changed hydrogeologic environment, which is affecting water quality and physiological response, and in turn affecting primary production, prey, and ultimately behavior and population. So early on in the project, from the literature and from beginning to develop a collaborative understanding of this ecosystem, we identified some things that we thought might be key indicators of cumulative effects. One was macrodetritus in prey production, export, and consumption. It had been hypothesized by Chris Sherwood back in 1990 that there had been a fundamental shift in the food web regime from a macrodetritus-based regime to a phytoplankton-based regime from the reservoirs. Another was connected channel edge development. So we know that the channel edge is really a nexus of terrestrial and aquatic productivity that's can be beneficial for fish feeding. And then the question of wetted area and inundation. This is a conundrum in the estuary because if you can imagine, if you pick any point in the estuary, water is constantly rising and falling. And it's doing it in a different manner every year, every day, every month. And these sites, all of these wetlands dewater twice a day. So your actual area inundated is constantly changing and very difficult to predict. And this is where some of Andre's work came in. So one of the first things we did was to try to set the program on a path by developing standardized monitoring protocols and data reduction procedures so that all the various coordinating partners would actually be producing data that could eventually be compared. And that was a success involving a lot of coordination. And eventually we produced a NOAA technical memorandum led by Curtis Regner that has those protocols in it. Okay, so that's kind of the framework. And what I want to talk about now is some of the findings at the estuary scale, so the lower Columbia River and estuary scale in terms of hydrology and vegetation, and a new proposed zonation. Just for context, these are the flow trends from 1878 to 2012. And you can see the overall decline in the peaks and the period of dam construction, and also that there's quite a bit of interannual variability historically and in the present. So the peak freshet has gone from something like 16,000 cubic meters per second to down to about half of that. So this is where it gets interesting, and this really points up the challenge of working in this system. This is these are all one-year hydrographs and from wetlands. And down here in Baker Bay near the mouth, you can see the dominance of the mixed semi-diurnal tidal signal. Again, you have 2 highs and 2 lows every day. Up here, just below Bonneville, you can see really the dominance of the fluvial signal where you have a winter peak and then an elongated spring freshet. And this is just an example of one site in the middle. These are our reference sites, by the way, our reference wetlands. you can see this, that it's a hybrid. You can see the winter peak, you can see the elongated, the bump that is coming with the freshet, but you can also really see the tidal signal. And so this is a gradient of change all the way along the river from this to this. And some of the factors are, of course, the tidal influence. It's very stationary signal down at the mouth because of the tidal influence, but there's also non-stationary processes from coastal upwelling and other factors. And then as we move upriver, we've found through a partnership with David Jay and his grad student Keith Leffler down at Portland State, they performed some power spectra, continuous wavelet transform, and harmonic analyses on our data, both the floodplain wetland data and the channel gauge station data. And they were able to identify thresholds along the way where there were changes in the relative influence of the seasonal patterns, the flow, interannual variability, of course, played in, and weather. So this is kind of part of the basis then that you can see again here this is up near Bonneville and this is down at Tongue Point, and these are all oriented from the Bonneville or fluvial side of the system down to the ocean side of the system. And what they're really looking for here is all the constituents that they're parsing the data by frequency to look at the constituents that relate to, say, biweekly tides. The fortnight, the 2-week is a big one. The monthly is a big one in tidal systems. The seasonal one. So they're parsing all the data from 1/400th of a day to many days to come up with these thresholds. So our part of this partnership then was really looking at the wetland data relative to what they were finding, and these plots are both oriented from river kilometer 0 at the mouth to Bonneville Dam at 234. And this is average site elevation of a wetland, and this is average water elevation, water surface elevation. And these data have all been corrected to the Columbia River datum. And the Columbia River datum is an interesting thing produced by the Army Corps of Engineers that can be very useful. It basically takes the longitudinal gradient of the Thalweg out of the data if you do the conversion. And so it helps us to look at things, sort of compare apples to apples from down at the mouth to up at the dam. And you can see really the narrow band at which these wetlands occupy, just a couple of meters. The bathtub ring around this estuary of wetlands is very narrow. And another point I want to make is just that these, of course, we can predict the location of wetlands from the water level in parts of the area that are outside the dikes, but behind the dikes, these relationships don't hold up. And these blue sites are tributaries. These are out on the tributaries to the main stem Columbia. So I don't have time to go into a lot of the data on vegetation plots today. We have over 4,000 quadrats that we've sampled. But I did just want to point out that the reed canary grass is a huge issue in this ecosystem. It's the red here. And one of the reasons it's an issue is that it extends all the way from 1.1 meter Columbia River datum up to the highest that we've sampled, which is about 4.5 meter. And it doesn't persist down in the saline areas and down in the lower elevation areas. So what we needed was a metric that would relate the plant communities to the water surface elevation regime. And I think Dave Merritt talked about something similar in his opening keynote yesterday morning, some exceedance value metric. And this is really just a simple cumulative sum of the difference between the water surface elevation and the land surface elevation at, in our case, an hourly time step, and for the range of the growing season is what we were interested in. So it's really just a cumulative sum of water depth during the growing season. And this turned out to be very predictive. This plot is river kilometer by meters CRD vertical by SEV. And you can see that as you go upriver, the SEV jumps rapidly. What we see upriver is months at a time early in the season because of hydropower system management where water is just sitting on these wetlands. And then in the lower river, we really have a much lower SEV where the tide is coming in and out twice a day. This SEV is predictive of the average percent cover at the sites, and we are actually developing a manuscript that has a lot of other analyses about the more detailed community things that I'm not ready to talk about yet today. But I wanted to talk about the large-scale trends. So these are our proposed zones then, and this is the SEV in meters per growing season that is characteristic of the estuary, the upper estuary, lower tidal river, middle tidal river, and upper tidal river. So in terms of plant species richness, we see some interesting patterns. Up here, where, as I said, there's a lot of water sitting on top of the sites, we see a diminished number of species that presumably are tolerant for that type of regime. Down here, of course, salinity influences what can persist. So we see a real high species richness here in the middle in the tidal freshwater zone. And for people who are interested in this, I find it fascinating. And there's this mid-domain effect hypothesis, and Sharp and Baldwin talk about it in a recent paper in Aquatic Botany. But these things, again, are important for planning all these restoration projects. We need to understand the hydrologic regime, and we need to understand what we can expect in terms of plant species in this highly diverse system. This is then an NMDS plot of a discriminant function analysis of the community data, and it shows it segregated into these 5 zones. And this is looking at the similarity analyses at the site scale, but what we're doing is averaging all the sites in one zone and their similarity to all the sites in another zone. So it's pairwise comparison averaged by zone. And what we see here is that there's the highest affinity or similarity to the sites within each zone. So each zone is less similar, the sites at each zone are less similar to the sites in any other zone than they are to each other. So these data then, from both the hydrologic data and the vegetation data, are really the basis of this proposed zonation. And we have a 2-part paper coming out this year. The first part is already out. The second part is in revision. And this is the proposed zone system that includes the hydrologic regime and this sort of large-scale vegetation transitions. And I want to make a note, while we're still talking about the estuary scale here, or the entire system scale, about sedimentation, because we have a real issue with sediment change here with the difference in energy in the system because of the management of the hydropower system. And so this is an example marsh today, and this is kind of extracting that to look at blue as open water and green as marsh. And this is what it looked like in 1878. It was all open water. So essentially what we found is that all of these marshes are prograding quite actively over the last 100 plus years. And similarly here, this is a more direct anthropogenic effect. This is a T-sheet from the 1870s where we have no islands here, but today we have these big dredge material deposits that have been placed there by the Corps of Engineers. So we're seeing really a change in terms of this filling in of the system. And what we've found then is that, what we've realized then is that all of our reference sites are really quite young. We did not know this at the start of this effort, but we've realized that these sites which the salmon will have access to that are out on the fringes are really, they've developed since the dams were put in. And what's interesting about it is that it gives us some hope about the responsiveness for under things like the climate change types of scenarios like Sarah talked about earlier. So really just a small percentage here in the dark of each of these sites was historically present. Okay, so now I want to do just a short dive into Sitka spruce swamp geomorphology primarily. So the problem statement here when we started this research, I guess about 10 years ago, was how do we restore a historically understudied ecosystem under a changed hydrologic regime? And the approach was to use multi-scale ecological research at paired restoration and reference sites to identify the controlling factors, the ecosystem structures and functions, achievable restoration targets, and appropriate monitoring indicators. So the question, why was it understudied in the first place, is that it was almost entirely eliminated. The estimates range from 77% to 90% that were converted. Basically, there was a lot of land conversion to ag, so folks logged the forest and diked the area and converted it mainly to dairy or other agriculture. But also, during the war, there was a U.S. Army Spruce Division that was sent to the Northwest that built the Spruce Railroad that you might have heard of and high-graded the spruce out of there. They took the biggest and best spruce and shipped it to our allies and to our own manufacturing for aircraft. So these are 3 of the remaining fragments. There's only several hundred acres on the Washington side of the Columbia, and these are owned by the Columbia Land Trust, Secret River, Crooked Creek, and Seal Slough reference. And this is one of the typical restoration sites here with some dike breaches and a culvert replacement. So starting at the reach scale, Our hypothesis was that the development of these tidal freshwater forested wetland channels incorporates large woody debris to form a low gradient step pool forest system. And we did just a simple longitudinal survey of the thaweg and the log jams and the pools to develop some data to analyze, to look at that. These are the results of the longitudinal surveys. The triangles are the log jams, and the black circles are the pools, which are in almost every case immediately above the log jams, and the gray is the substrate level. And there was no significant difference between these three restoration sites in channel widths per pool. And so working with Dave Montgomery, putting this into, looking at this relative to other data on four-step pools and channel classification, it was squarely within the four-step pool channel type. So we published that classification in Restoration Ecology in 2009. So secondly, kind of expanding out to the watershed scale, I want to talk for just a minute about hydraulic geometry for those of you who haven't studied it. Back in the early part of the last century, when engineers were trying to design navigation and keep tidal inlets stable, they were really looking at these relationships between cross-sectional geometry at the outlet and the size of the tidal prism. Later on, Leopold and others did similar work in fluvial regions, looking at channel cross-sectional geometry as a function of flow or Q. And in fact, this paper, although they don't really say it, I am quite sure was in a tidal freshwater system. So more recently, we've seen people in restoration ecology develop surrogates for Q in salt marshes. So tidal prism, catchment area, and total length of tidal channels have been used in the San Francisco Bay Delta and for the United Kingdom. And so the null hypothesis here was essentially that none of these relationships pertained. There was no relationship between channel cross-sectional geometry at the outlet and any of the catchment scale metrics that we could measure for our system. And the methods here involved Andre Coleman doing some extraction of the channel network using methods that were sensitive to the fact that we had forest cover and wetland, which are two of the most difficult things in remote sensing. And then me going out and surveying all these cross sections, and we developed a blind test where I went out and ran blind transects also to verify that his channel network was correct. And we put those data together. Again, we just have these 3 points, not the strongest, but this is catchment area versus cross-sectional area at the outlet, catchment area versus channel length, and then total channel length versus cross-sectional area at the outlet. And again, because we just had the 3 points, I found all the data that I could where people had actually published tabular data and plotted our points, which are the stars here, with the San Francisco Bay Delta data and with the United Kingdom data. And it's not bad. They're within the cloud, which is pretty much all we could hope for, considering they're further up the elevation gradient and in a different part of the world. And we published this in a Polish journal, Ecohydrology and Hydrobiology, in a special issue from the UNESCO Floodplain 08 conference. So another part of trying to identify these reference targets then is really getting a clear idea of the elevation range of each of the individual plant species. And we've done this for every site we've worked at, but I just wanted to highlight it here for the spruce swamps. And so far the species richness that we've found here in the swamps is 42 herbs, 22 shrubs, and 9 trees. But we haven't been able to work in the early spring, so there's probably some additional herbs out there. So now I want to just talk about a couple of the restoration site findings relative to the reference site characteristics that I just mentioned. Here is a typical reference site. This is a good-looking spruce swamp channel to me. It's got a nice nurse log across, channel spanning nurse log. This is low tide. At a higher tide, the water would be right up to bankful width here, just below this nurse log. These are typical restoration sites in the very early stages of restoration. So here you have the pasture that you can just glimpse in the background, and these were a few trees that had taken hold since the area was diked. You can see now their feet are pretty wet and they're dying. And this is a typical dike breach in the early stage, within a month. So we looked then at the restoration sites, the pools per 100 meter, versus the reference site condition. It was significantly lower number of pools. So we looked at whether or not there was going to be enough wood in the system to create the pools based on this four-step forest step pool channel type, large wood forest step pool channel type. And we did find that there was some fallen wood being recruited in through the fact that these trees were dying with the inundation, and also some legacy wood being exposed as the fine silts were being moved out, but it was nowhere near the quantities that we saw in the swamps. Then through Andre's work, we looked at the microtopography and established how much higher, how much coarser this was in the reference site. This is an example reference site here and the restoration site here. And furthermore, it was quite compacted and subsided. So it was on average about 0.7 meter lower in the restoration site than in the reference site. So really we're seeing the role of large wood in producing this kind of hummocky swamp microtopography, which is also substrate for tree reproduction. We do in fact have a lot of spruce reproduction going on in our swamps. And yeah, so they're topographically rough but subsided. So topographic evolution then, we looked at accretion rates from 2005 to 2009 at 12 points in the swamps and 5 points in the restoration sites. The mean in the swamps was a half a centimeter per year. In the restoration sites, it was 2.3 centimeters per year. And that was significantly different. And this is where it gets into the kind of the detective work of putting these things together. So this is what we saw at restoration sites. This is the catchment area versus cross-sectional area at outlet that I showed you earlier on a different scale. So this is that reference line with the 3 swamps that we surveyed. And what we would expect in the idealized world of the Bradshaw model or any kind of restoration and reference framework is that our restoration sites would be trending toward the reference sites. In fact, what we saw, the X's here are the before and the triangles are the after. So this is one that trended way away, and that's this one here. This is 4 years of cross-section surveys at the outlet. This one here actually crossed the line and kept going. This one trended away also in a small level. And so we finally realized that, in fact, the subsidence and lower area of all of these areas behind the dike had produced this much larger tidal prism. And because channels exist to convey flow, they needed to be much larger in the restoration sites than they were in the reference sites. And so we started trying to estimate, based on the sediment accretion data, how long it might take for the site elevation to reach the levels of the spruce swamps, and it was somewhere between 20 and 54 years. So Andre developed, in order to try to help us scale our point water surface elevation regime data up to the sites, Andre developed this area time inundation index model, and it integrates terrain processing with terrain analysis using deterministic infinity method to be, both of which end up being very sensitive to these very low relief areas where literally centimeters can matter in your plant community or your plant association, with an inundated area algorithm that he wrote to propagate water over the landscape, starting with the kernel where your pressure sensor is located. And this is in review at Ecological Engineering right now. So you can see, for example, this is just a animation of how the water is being propagated. But we can actually calculate through this model at this point. It's in Python. It's very adaptable, 14 different spatial metrics and 49 tabular metrics that are really useful. And so, for example, this kind of corroborated what we were thinking about what was going on with these channel cross-sectional areas, because the frequency of floodplain inundation at the reference site was 18% compared to 54% at the restoration site. So finally getting to ecosystem function then, and juvenile salmon and prey resources. In insect fallout traps, we found that about half of the taxa present in the spruce swamps were also present in the salmon guts. And in electrofishing effort, we found juvenile Chinook salmon, chum salmon, coho salmon, and cutthroat trout, as well as Pacific lamprey. And there wasn't a lot of difference in the mean taxa in the restoration and reference sites. So now I want to talk just, while I'm on the spruce swamp topic, I want to talk about synergy just a little bit, getting back to cumulative effects. This is an effort that we undertook to look at the potential for this effect of high-density clusters of restoration. And we divided this Grays River landscape into downstream, midstream, and upstream reaches, and went to the historical photos and T-sheets to find that there had historically been 42 little channel openings there. And so we viewed this as the population or the universe for a statistical model from which we drew randomly selected sets to breach in the terrain and then run a hydrodynamic model of. It was a FVCOM model. And so this is a test of this particular hypothetical relationship. And these are several hypothetical relationships for cumulative effects that we put in our approach paper back in ecological restoration, but hadn't been able to test yet. And so we're really looking at whether the cluster size of restoration projects affects the mean response per project. And these were the results. Again, we did segregate into downstream, midstream, and upstream reaches. And so first, this is the spatial configuration effect, where we have the average wetted area per breach being much higher for the downstream breaches than the midstream breaches or the upstream breaches. And then the proportion of channels opened to the proportion of area wetted shows a distinct nonlinear effect up through about 25%, where we have this synergistic effect on the proportion of area wetted, and then a steep diminishing return. Okay, so just to wrap this, this section up then, and this is the kind of the implications for spruce wetland restoration planning and evaluation from this research. First of all, spruce swamps are salmon habitat, and this is something that came as a surprise to a lot of estuarine ecologists who were used to thinking that marshes were salmon habitat, not forests. But it's very much in line with, for example, I know a lot of you have cited and worked with Michael Pollack's work. It's very much in line with that, with where we have these coho producing pools. Secondly, controlling factors and restoration targets can be identified from reference sites that are subject to the altered Columbia River hydrograph. Also, prior land use shapes the restoration trajectories of the channels in the plant community. So the sediment budget and the sediment accretion rates are really important indicators. Inundated area is super likely to change as the land is accreting, yet the project sponsors are requiring, the funders are requiring project proponents or the implementers, the practitioners, to report based on a per area basis. That's not a stable metric. Large wood is important to pool formation in tidal systems, but there's not enough available in these diked areas to actually produce those pools. And finally, this space crowding, this high-density cluster of restoration actions can produce synergistic effects in the wetted area per dike breach. Okay, so now I'm going to shift back to concluding the evidence-based evaluation of the early stage Columbia Estuary Ecosystem Restoration Program. Last 10 minutes. This is a map of the Lower Columbia River and estuary showing our study sites coded, not all of the reference sites, but the ones from which we drew specific data for each of the 5 lines of evidence that are located in the floodplain. And we also pulled data from salmon stomachs up at John Day Dam at river kilometer 349 and from Bonneville. I want to start with the cumulative net ecosystem improvement model. The NEI model was first proposed by Ron Thom in 2005, and we put a simple equation for it into our 2011 paper, which is simply the change in ecological function times the project size in area times the probability of success, which is presumably going to be less than 1. And to scale it up, we took this for the whole universe of restoration projects, 1 through N. So we applied this model for primary and secondary productivity metrics in the ecosystem. And these data are drawn from everything we can get our hands on, master's theses, published literature, anything peer-reviewed. And this is river position, so in terms of the zones that I presented earlier, so this is the downstream zones, the upstream zones. There's no data from higher in the river. Like I mentioned earlier, there's been very little study up there, but we're trying to get some data on that now. And then this is the number of sites, and then the number of samples, and then the mean, dry weight, and the standard deviation. So we do see a lot of variation, but the mean values are 600 to 1,125 grams dry weight per meter squared for the reference marshes, and getting close to that for the recently reconnected marshes. As part of this research, we also, this is back to the Grays River, developed a particulate organic transport, a particulate organic matter transport model, and parameterized it with biomass, above ground biomass data that we had clipped in summer and winter. And so it's really just an above ground effects model. But it showed that the matter could be transported, in this case 7 kilometers out to the main stem river to augment the food web, and of interest from a cumulative effects perspective. It also was transported onto other restoration sites owned by the Land Trust in this area. This has the same format, but it's the secondary productivity data. So again, this is river position, the number of studies and samples, and then the prey, the chironomids, the other dipterans, hemipterans, arachnids, and amphipods. And this is where we get into some challenging area in terms of scale effects, because obviously all this sampling is at the 1 meter scale. And really what we're trying to do is scale up to the kilometer scale and multiply these things out by the total area restored in these different reaches. The numbers are coming out in billions. We probably shouldn't have a 0.3 here. It's probably that coarse in terms of the effects of scaling on this. But we can say that there are a large number, that there's a lot of macrodetritus being pumped into the system, and there's a lot of prey being produced in these now hydrologically connected areas. So another line of evidence then was the analysis of time lags at historically reconnected sites. And we had hoped for a robust chronosequence, but we could only really validate the dates of natural breaches through storms well for 3 sites. So one had been 60 years since breaching, one had been 50 years since breaching, and one had been 10 years since breaching. And we did a rapid assessment of those, and I don't have time to show the data today, but the short story is that all 3 are marshes. So if trends are going to continue similarly to the last 10 to 60 years, which is in question with climate change, this is a positive result for marsh development. This then is site scale process response, and here you see the water surface elevation regime at a reference site versus a restoration site before the dike was breached. It has an immediate response where they come into line. And here you have the sediment accretion rates for 3 paired restoration and reference sites. And the basic take-home story here is that the restoration sites are all lower elevation because they're all subsided behind the dikes, and they all accrete faster. This is the analysis of salmon stomachs, and this was several thousand salmon stomachs from the estuary at river kilometer 15, Bonneville Dam at 236, and John Day at 349. And this is the percent that are actively feeding. So this is based on the percentage of stomach contents that were identifiable. And because of the evacuation rates of a salmon, we can expect that this feeding of identifiable prey in the stomach was probably within the last 30 hours. And so we're seeing that there's not a lot of feeding up here at river kilometer 349, maybe 10 to 20% active feeding, very little when they exit effectively the hydropower system. And this has bumped up to 50 or 60% in the lower estuary. What we would hope is that if somebody does this 10 years from now and there have been thousands of acres or thousands of square kilometers restored, that this number is going to get even higher because there's room for growth here relative to other estuaries in the region. And I've been told by people at the National Marine Fisheries Service that this graph has had a tremendous impact on interior basin fish managers, because for decades they've really thought about barging the fish out through the estuary. They haven't really recognized that the estuary could have a positive effect on these interior basin fish, these yearling Chinooks. These are fast migrating fish. Some of them go through the estuary in 3 days. This is then a meta-analysis of the action effectiveness data, and the framework here is sufficient evidence to conclude a causal correlation, suggestive but not sufficient, inadequate, or suggestive of no causal relationship. And here you see that all of these Ds are in tide gate replacements. This has had an effect already through the adaptive management project on the program, where they are they're not funding tide gate replacements anymore because of these findings. But we see that for the other methods like grading and dike breaches, we're getting very strong, sufficient data for the fast response variables like water surface elevation, and suggestive data for slower response variables. This then is integrating the analyses, all the analyses for each line of evidence by the fish-based responses and habitat-based responses. So these are the ancillary hypotheses again. And this is where I think that this framework is pretty robust, because each ancillary hypothesis, each metric is being tested by multiple analyses which are tied into these lines of evidence. Again, the only things that are suggestive of no causal relationship is the tide gate replacements, and we're seeing some positives in some things that are actually sufficient and others that are suggestive. And I don't have time to go into the detail because I'm running out. So what I want to say then is we rolled this up then through the causal criteria framework and found that all of these causal criteria for associations were supported, not for tide gates, but for dike breaches, except for experimentation, which wasn't evaluated because there were no formal experiments, and predictive performance. Like I said, we're not at the modeling stage yet with this ecosystem. Hopefully we will be someday. So the general findings were that this restoration supports increased juvenile salmon growth and fitness directly when they access the sites and indirectly when through allochthonous supplementation. But I didn't show these results, but we do have cumulative impacts of urbanization and forest cover loss in the watersheds. And we do see nonlinear, synergistic, and pulsed effects. And these need to be considered. These effects are complex in space and time, and so we need to consider them when we're doing predicting the trajectories of both the system and the benefits to salmon. So we did conclude then that there was the primary hypothesis was supported for this early stage. There's a lot more data that would be needed in 5 or 10 years, and the beneficial effect is expected to increase over time. We're reasonably certain about the positive direction, but not certain about the magnitude. There's not enough quantitative data. Some of the things we're lacking are the rates for the relationship between primary productivity and secondary productivity, especially what's going on with these reed canary grass dominated areas. Juvenile salmon growth rates, it's really hard to measure in situ growth rates at a site that dewaters twice a day. And then the need for experimentation. As a CODA, we did a little bit of study on climate change effects, and this is going to be what needs to happen next because the bathtub ring is going to go up. We're predicting significant increases at Astoria over the next 50 years, and these wetlands are going to have to migrate. So with that, thank you very much. And I'd like to acknowledge the Corps of Engineers and Bonneville Power for the funding support for the study. Yeah.
Is there any way to take a longer-term look at the salmon growth and look at the difference in growth rates and maybe survival between the fish that have the estuary and those that are barged through?
Yeah, I don't know of any barging going on right now, but that is really it's called the complete life cycle study. And that's sort of the holy grail. I mean, it would be great to be able to do that. One of the issues here is that it's a technology issue, actually. It's a tag size issue. And so the yearlings tend to, going back to the life history diversity, yearling salmon tend to migrate out faster. They're ready, basically. The sub-yearlings tend to make a lot more use of the estuary, and we don't have tags of a size and a life yet where we can actually tag them and know who they are when they come back. But that's being worked on. So, good question. Yeah, that would be a nice, simple technology fix. So, yeah.
Do you have any idea what the residence times are for salmon in the estuary?
There's been some work on that. That's been more targeted site-specific work where they used acoustic tags at the Sandy River Delta, and they found fish there up to 6 months, I believe. So, but it's really variable. There are actually also Lori Whitecamp's work on purseine data at River Kilometer 15, where those stomachs were from. Excuse me. She also has looked at residence time a bit from, I can't remember how though, but there is a study from NIMS by Lori Whitecamp that talks about it a little bit. But what we do know is just that the sub-yearlings tend to reside longer, but there are resident strategies. There are strategies where fish go down and hang out in the estuary for a substantial period of